as one of the organizers of this conference, I'm chair of the ASAGI Lab. Thank you for all of you and then coming participants and then presenters. Because since we knew that the Singapore attracted talented scientists and then like a student, so we from Japan, we are very interested in like working or launching this kind of conference. So I'm very happy to having this conference. Yeah, so this is the picture, and then hopefully next year also have this kind of conference in Japan. And then yesterday, Professor Kennedy mentioned that the very interesting the longevity, lifespan expectancy, and health lifespan expectancy difference, especially in the U.S. The gap life expectancy and health life expectancy is increasing, and then now the 12.7 years difference in the U.S. But in the Singapore and Japan, healthy life expectancy is very short than other U.S. countries. And then I think that almost everyone misunderstanding that in Japan, which area is longest lifespan expectancy. Everyone thinks Okinawa, but as you can see here, Okinawa is almost second or third worst the life expectancy in Japan. And then top is Shizuoka Prefecture. And it's two years difference. I guess in the Okinawa, the food, the environment, or behavior environment, behavior have been changing because of the U.S., the culture, changing the Okinawa's health. But the aging is so complicated, as you know. It's not the linear model. Maybe it's a non-linear model. Everyone changing the behavior or diet. Even like this is a mouse model published to 2020, and then some of the gene expression is keep increasing, but some gene expression is increased, but sometimes down regulation and increase. So this kind of the aging is not simply it's a linear model. It's a non-linear model. So it's very complicated. But to understand this kind of the mechanism, many of the people have been using the O-link or SomaScan, trying to isolate many of the peptide and then associated with the phenotype or diseases. Even in the hospital, large language model, health check record, which is language, and then about 10,000 the patient data sets can predict their death rate. So the very large scale of the data is very important. And then benefit or strength in Japan is, as the Dr. Okano presented yesterday, we have the super centenarian or centenarian cohorts. And yesterday, the Okano sensei mentioned the young centenarian also we have. And then, and then in this audience also like a Meiji or Morinaga or some of the maybe it's a Haichu you maybe tried before, but the Japanese the prefecture or company can make stuff oishi, very sweetie, and then this is kind of like this is say the active nutrition. Active nutrition is actively the sensing the aging clock and as well as the intervention with the food or other stuff. And then in Japan, the animal sample also very easy to access. So because of the zoo or the aquarium, many of we can easy to communicate and get many of sample. Today, I'm very interested in salamander talk, but and then I have been working on the shark and then giant squid and ayu, those animals as well. Today I have limited time, so I don't speak about. But this shark has about 400 to 500 years old, the lifespan expectancy. And giant squid has the body size of about 15 meter, but life span is about 3 years. So very diverse aging happen in the different animal model. So to accelerate aging, the research or translation, we developed the ASAGI project in Japan. And then this is the entire ASAGI project, and then the non-profit organization and the profit organization. And then non-profit organization will provide non-equity grant and for young scientists, and then collaborating with the local government or Japanese government as well. And then Nakanishi Sensei is the one of the trustees. And then ASAGI Lab, this is already public, but we are collaborating with Morinaga and other companies as well to accelerate aging the product. And also we launched the ASAGI Venture Capital, which is we will invest the new idea. And then in this audience, we have the general partner here, Kurihara. So I'm happy to you guys to talk to him. But I'm not going to talk about all of the ASAGI project that since this conference is focusing on mental health and then neurodegeneration, the experiment. So I'm briefly to introduce what I'm doing in the laboratory as well. And especially one of the topic today is the non-visual photoreceptor of the OPN5. Now there is maybe if you go outside, you can see the green, blue, or the red, many of the different colors, which is detected by visual photoreceptor. But actually there is another photoreceptor named non-visual photoreceptor detecting the violet light and the blue light, named the OPN5, OPN3, and OPN4. And especially OPN5 is located in a ganglion cell. The previous and then OPN5 is conserved among a lot of the species. For example, lion or many of the species can see only the red or green or a single color, but the OPN5 is conserved among the species. So maybe there is something very important, the function. And then actually the previous paper said that OPN5 is regulating the circadian rhythm, but I don't think only the circadian rhythm is very important. And we have been checked whether the OPN5 is expressed in the mouse retina. And then there is the OPN5 expression, the retinal ganglion cell, and OPN4 expressing cell. So meaning that even the non-visual photoreceptor blue or violet light are sensed by the different way or different the cell. And then OPN5, the question is where the OPN5 signal, violet light signal from the retina is going to the brain. So this experiment show that the OPN5 signal, violet light signal going to the medial habeneural region directly. So not going to the visual region of the brain. The violet light is directly going to the medial habeneural area. So different the circuit. And interestingly, by using the SDS mouse model, this is the social defeat stress model, the depression. So we induce the depression. And then by induction of the giving the violet light in the animal room, the depression is recovered. So violet light is very important to improve the depression, and which is depending on the OPN5. So everyone, I think, maintaining the animal in the room, but in this circumstance, there is no violet light because LED provides blue, red, and green. There is no violet light inside. So maybe in the circumstance in the animals may be depressed. So please give the violet light. And then, interestingly, there is the astrocyte, microglia, and then oligodendrocytes in the brain. And then violet light activate the oligodendrocytes specifically. So oligodendrocyte may improve the depression function through the oligodendrocytes. And we developed, but in Singapore, maybe it's very hot, humid. So even I say the violet light exists outside environment, sunlight. But maybe you don't go outside because it's hot, humid. So that's we developed glass with LED. So you don't need to go outside. Just wear the glass. Then we tested the human clinical trial for depression patient, which is a crossover, the design. And then violet light glass actually improved the depression patient in MADRS, MARDS score. So the violet light actually works in human. But not not only the depression, we give the violet light in the dementia, the mouse model as well. And in dementia mouse model, by giving that violet light, the oligodendrocyte-associated genes are upregulated. So in the depression mouse model, the oligodendrocyte is important. And then in the dementia model, the spatial cognitive function is improved. And in human, the violet light and the violet light with 40 hertz, I don't deeply go the 40 hertz, the frequency, but whether the violet light change the brain wave in the human or not in the human clinical trial. And then the alpha gamma, the coupling is improved in the violet light. Then, of course, if the oligodendrocyte is involved in the brain function, the oligodendrocyte is associated with Parkinson's disease. So we actually conducted the Parkinson's disease clinical trial as well. Then in the Parkinson's disease patient, hallucination is very important for the Parkinson's disease patient, and which is improved. So go outside, or get a glass with a violet light. Another topic I would like to share is the epigenetics memory or postnatal regulation of the aging. So recently, epigenetics memories happen if you get a high BMI or infection happened and a long-term effect. And then previously, I have been in David Sinclair's lab, 2013 to 17. I left his lab, but after 8 years, after 6 years, we published a paper in the ice mice, and there's some of the conflict, but I don't talk about some conflict. But this mouse model show that the short term of the DNA damage accelerate the change in epigenetics and long-term effect in the aging phenotype. And then by using this mouse model, what kind of gene expression is changing, or even human, the tissue sample, the database, GTEx, maybe some of you have been using. And then our lab also try to isolate what type of the transcription factors can rejuvenate the aging phenotype. And then using, this is unfortunately linear model because of the limited number of the sample. But for example, in the brain, the RAX1 and BRD2 and NANOG is one of the top transcription factor to regulate the aging, could be. And then breast is a TP53 and STAT3 and HDAC. And then by combination of the data sets, we try to isolate what type of the drug can rejuvenate or reverse aging phenotype. And end up, we isolated the ambroxol. Ambroxol is a well-known exporterant. So if you get cough, you get ambroxol. And then ambroxol is well known to target recently the GBA1. GBA1 located the lysosome and activate mitochondrial lysosome or autophagy. And in the US, the ambroxol is now under the clinical trial for Parkinson's disease. And then we use this ambroxol for the aging phenotype, the grip strength or cognitive function or other flail. And then ambroxol treatment increase the muscle function in aged mice. And then increase the muscle, the diameters, and then improve some of the gene expression, FOXO or other expression pattern. And not only the muscle function, ambroxol treatment in the aged mice, improve the cognitive function. So maybe in Japan, the ambroxol is popular, the drug that the aged people take. So that could be the Japan has a long lifespan expensive. But anyhow, the fertility index also improved the Ambroxol. So based on these data sets, we started a clinical trial. So we applied XPRIZE hairspan. XPRIZE hairspan is to aim to reverse aging phenotype at least 10 years, and maximum 20 years we aiming to. So we designed a clinical trial, and then as the mentioned before, maybe the combination is very important. So in this clinical trial, we mixed exercise. So exercise or only the drug, or exercise with drug. And then we are going to test this kind of the biomarkers or like a physical test. So we're gonna get the data the next year. So in the ASAGI lab and my laboratories, Japan is very unique circumstance. So I hope that in the future, in the Singapore, the scientists and the Japanese scientists collaborate, or we can have to start the exchanging program of the young scientists. And then aging is very complicated, this system. So not only the understanding aging mechanism and a non-linear model, but also even like aging clock have been developed. We have to test by intervention how this intervention improved the aging clock. And then delicious or happy is very important. So even something like a very impact on our health. I don't want to eat very like a non-delicious food or stuff. So happy is very important. And then diverse of the aging. Recently, the whale or naked mole rat or many of the diverse, the animal has a different function of the gene or proteins. So those the diverse aging process is also very important. And then so this is the acknowledgement of my laboratories, and then thank for the David Sinclair's lab. And then next year, I hope that I can talk more about diverse. This is the best interest, so not human. I like the animals. Yeah, so thank you for the participate this conference, and hopefully, yeah. Thank you very much.
Questions?
Dr. Hayano, a really interesting talk. I was especially struck by your work on OPN5, because immediately I'm starting to think about the seasonal affective disorders, right? How a lot of people, they do get more depressed in winter, and then they do feel a little bit better once spring starts, and of course that might be related to the UV accumulation that they are getting. So I was just wondering, along the same lines, have there been any work on whether OPN1 possibly have a polymorphism or a genetic mutation that predisposes to depression? Any work along those lines?
We haven't done that experiment, but of course the COVID-19 situation, the people stay house, and that could be the impact on the health. And also, if you wear the contact lens and then the glass, almost like 100% block the violet light. So we should not wear the sunglasses or like the glasses. But this idea came from the myopia. So myopia is a big issue in Singapore as well. And then outside of the environment, on the OPN5 and the violet light actually improve the myopia. So outside the environment is very impact on our health.
Thank you.
Yes, thank you very much. So, yeah, I'm very surprised that the Shizuoka prefecture has longest life expectancy. Do you know there's east side and west side of Shizuoka has a difference? You know?
Yes.
Yeah.
Interestingly, the east side of the Shizuoka longer life expectancy,
really?
And then west side shorter.
Yeah, it's important to know that the east side of Shizuoka medical service poor, very poor. There's no university hospital. University Hospital only in the Hamamatsu,
Yes.
the west side.
Yes.
Even that, so
Yes.
eastern parties. So maybe so environment could be different. Yeah.
Yes. So
Yeah,
the
yeah.
hospital system may not be the important for the life expectancy.
Yes, yes.
And then the east area, I'm not sure that works or not, but the east area eats, drink the green tea, and then the water, the component also are different. So we are looking for what is the mechanism or cause the east coast of the Shizuoka Prefecture.
Mount Fuji.
Yeah, Mount Fuji may. Yeah, excellent lifespan.
So sorry, local question.
We still have time. 1, 2 questions?
Beautiful talk. So what's the mechanism of action of ambroxol then? How is it affecting all these different aging parameters?
At least so far, based on my experiment, mesenchymal cell, the differentiation and proliferation was affected by the ambroxol. Maybe it's because of the protein homeostasis, but in the muscle, there is the mesenchymal-like cell, which is the fat adipogenic progenitor cell, which is an impact on the ambroxol.
Okay, thanks.
Okay. Thank you, Prof. Hayano.